Do you find yourself struggling with numerical problems in biochemistry? Fear not, biotechnicals. Today I am here to equip you with the basic concepts for numerical problems in this subject. Whether you are a student preparing for the upcoming CSIR NET Life Sciences exam or any student trying to solve experimental calculations in your lab, this video is designed to make these concepts very familiar to you. Let's dive in. I want to remind you that these concepts will be helpful in other entrance exams as well, such as GATE, BT, Excel, ICMR, TIFR, ICAR and also DBT, because these exams also include numerical problems in biochemistry. First concept is regarding the mass of a substance. If you talk about a simple substance, when you try to tell about the mass, you have two things associated with it. One is the value, numerical value. In this case, it is 2 kg of mangosteen, 3 kg of papaya and 50 grams of flour. And the second component to it is the amount. That means the unit. So 2 kg, 3 kg, 50 grams. Don't just look at the numerical value. The entire amount will be dependent on both this component. As 3 and 50, between these two, 50 is a higher numerical value. But then 50 grams is overall a smaller value because of the unit. Gram is much smaller than kg. Similar to the amount of mass, we have something called as molar mass. This is your first concept for today. We will understand what is the meaning of molar mass and also what is the relation to Avogadro's constant. Molar mass is nothing but mass of 1 mole of element or compound expressed in gram. The unit of molar mass is grams per mole. That means how much grams will be the weight per mole. Now what is 1 mole? As I said, mole is related to the Avogadro's constant. 1 mole, molar mass is equal to 1 mole, mass of 1 mole. And 1 mole is just a number. What number? It is the Avogadro's number, which is equal to 6.02 into 10 to the power 23. This can include any particles. For example, if you take 1 mole of atom, it will equal to 6.02 into 10 to the power 23 atoms. Similar to that of a dozen, as we say in a dozen there are 12 particles. Similarly, 1 mole is always 6.2 into 10 to the power 23 particles. Then what exactly is molar mass? It is the mass of 6.02 into 10 to the power 23 particles. Thus, if you take molar mass of one atom, it would mean mass of this many atoms. If you take molar mass of a molecule, then it would mean mass of these many molecules. Now, exactly how much is the molar mass? For this, we have to refer to the periodic table. When we refer to the periodic table for a certain element, let's say carbon, we know that the atomic number and the mass number is mentioned for that particular element. Like carbon has total of 6 protons and total of 6 neutrons. However, electrons is not mentioned in the mass number, only proton and neutrons are mentioned. So 6 plus 6, 12 is the mass number of carbon. But this mass of carbon mentioned in the periodic table, it is only for one atom of carbon. Now if we take 1 mole of carbon, that means we haven't taken 1 atom of carbon. Instead, we have taken 6 into 10 to the power 23 atoms of carbon. Now imagine if 1 atom of carbon mass is 12, the unit for this is AMU, which is atomic mass unit. It is also similar to that of Dalton. Now what is Dalton? Dalton generally we use for molecules, the mass of molecules, and atomic mass unit we generally used for atoms. Okay. So atomic mass unit, if you want to express it in gram, 1 atomic mass unit is equal to 1.67 into 10 to the power minus 24 grams. Okay. So if carbon has total of 6 particles, protons and neutrons both are equal to 1 AMU each. So 6 into 1.67 into 10 to the power minus 24 into 2, because neutrons are also there. So basically 12 into 1.67 into 10 to the power minus 24 gram. This is what is the mass of 1 AMU. So instead of writing 1 AMU, the grams every time, we have to write it in the form of AMU. Okay. So if we have one atom of carbon for 12 AMU, then 1 mole of carbon, that is 6 into 10 to the power 23 atoms. I hope you understand 10 to the power 23 is how much? So 10 followed by 23 zeros. You can well understand how much big this number will be. That many carbon atoms, obviously the mass is going to be more. But how do we have this mass? We have the same numerical value that we have from the periodic table, just that instead of AMU, we will be writing grams. That means 1 mole of 6 into 10 to the power 23 atoms of carbon will be same as the atomic mass, but in grams, expressed in grams. Similarly, if you take for sulfur, the mass number of sulfur is 32, which means that if you take one atom of sulfur, just one sulfur atom, the mass will be 32, the unit will be AMU. But if you take 1 mole of sulfur, which means you have taken 6 into 10 to the power 23 atoms of sulfur, then that will be equal to 32 grams, right? That is how molar mass of an element is found out, by looking at the atomic mass of the element on the periodic table. So we have on the periodic table, considering all the isotopes, we have something called as relative atomic mass or relative molecular mass. If you can see all these components which we have taken, whether we have taken sulfur, copper or water, all of these are having 1 mole. Okay, why? Because they all have 6 into 10 to the power 23 particles. Okay, but their mass is different. Why? Because these are different elements, right? So elements and compounds. So their mass will be different, and how much of the amount will be present. So you can see 1 mole of water is only this much, but 1 mole of copper is this much. Okay. So this is how all of these substances, they are containing 6 into 10 to the power 23 particles. Now one example, 1 mole of aluminium, how can you write it? 1 mole of aluminium has a mass of 27 grams, or we can also say 1 mole of aluminium contains 6 into 10 to the power 23 atoms. Now you can combine both of them, and you can also say that 6 into 10 to the power 23 atom of aluminium has mass of 27 grams. Okay. Next is calculation of molar mass of a chemical compound. Molar mass of chemical compounds are nothing but equal to the sums of the molar masses of all the atoms in one molecule of that compound. For example, if we take NaCl, we have 1 mole of sodium and 1 mole of chlorine in this. So the molar mass of sodium chloride will be molar mass of 1 atom of sodium and molar mass of 1 atom of chlorine. So we all know that the sodium, 1 mole of sodium will be 23 grams, because 23 is the mass of sodium, 23 AMU. If we take 1 mole of sodium, it will be 23 grams. And if we take 1 mole of chlorine, it will be 35.5 grams. So total will be 58.5 grams. Okay, so if we take 1 mole of sodium chloride, 1 mole of 58 AMU if you take, then this is the mass of 1 molecule of NaCl. Okay, 1 molecule of NaCl. But if you take 1 mole of NaCl, then you are taking 6 into 10 to the power 23 molecules. So the mass will be 58, but expressed in grams. Okay. So like this, you can pause the video and find out what will be the molar mass of water. Here is the answer for this. Water contains 2 moles of hydrogen and 1 mole of oxygen. 1 mole of hydrogen is 1 gram. If you take 2 moles of hydrogen, then it will be 2 grams plus 1 mole of oxygen, which will be 16 grams. Total molar mass of water will be 18 grams. Third concept here is solutions and dilutions. When we have solutions, we actually prepare some solid particle with some liquid component. Most experiments involve at least one solution. A solution contains one chemical dissolved in another liquid. When chemicals are in the form of liquids, if both are in the liquids, they can be either pure liquids or pure solutions. A pure liquid will be something like absolute ethanol. It contains nothing but molecules of ethanol. When dealing with solutions, the chemical which is in the smaller quantity or which is the solid component, it is called as the solute, and the liquid it is dissolved in, it is called as the solvent. So when solute and solvent combine together, they make solution. If the amount of solute dissolved in the solvent is calculated, then the concentration is determined. That is our next topic, which is finding the concentration of the solution. All this time I was telling you the molar mass, which means what is the amount, but right now we will be focusing on how to find the concentration. Before going into that, let me tell what is concentration. Concentration are terms which are used to describe how much solute is present in a solution. Which means if you are supposed to express only the mass, you will use a single unit. That means grams. We were using grams or AMU or Dalton. But now that we have concentration, you have to say 2 units. That means the unit of the solute, which can be gram, and the unit of the solution, which can be liter or milliliter, anything like that. Okay. So it is important to be able to distinguish values that are concentrations from those that are not. So those which don't have 2 units, only 1 unit, that we were calling it amount. Learn about the technical word also. If we put 1 gram of salt in a beaker, that it is an amount. If we take only 1 gram of salt, it is just amount. But if we add 1 liter water to that salt, then it becomes a solution. And now we will have to mention the concentration. After adding the water, if we still say 1 gram, it is not correct. We can say that it is 1 gram in 1 liter of solution, which is described using values for concentration. Similarly, you can take any other unit for the amount. Not necessarily always you have to express in gram. So you can take 1 mole of salt also. So 1 millimole of salt or 1 mole of salt in a beaker. Again, it is an amount. Now, if we bring up the volume to 1 liter with water, then we have 1 millimolar solution, which is also described using values for concentration. I will be telling you. A very important point to be considered here is that amounts are always additive, but concentrations are not. Which means if you add 1 gram of salt from one beaker to another 1 gram of salt in another beaker, 1 plus 1 will give you 2 gram of salt. So since these are amounts, these can be added. But if we take a solution that is 1 gram per liter in a beaker to 1 gram per liter in another solution, then we will not produce 2 gram per liter. Because now since we have 2 liters, we have 1 liter plus 1 liter, we have 2 liter, and on top also you are adding, so you will have 2 grams per 2 liter. 2 gets cancelled, and what you actually have even after mixing is still 1 gram per liter. So directly you can't just add up the concentrations. This is a very important point. If you miss out on this, there can be mistakes in your calculation. Now coming to the next concept, which is molarity. Molarity is a concentration term which you use for solution. This gives the moles of solute in 1 liter of solution. As I said, for concentration you have to use an amount of the solute and in some amount of solution. So you need 2 units for that. Which means molarity is nothing but moles of solute divided by liter of solution. Make sure you are always using this concentration in liter. The unit should always be liter. However, on top you can either keep millimoles or you can keep moles. Accordingly, if you are using moles, you will be getting molarity, which we represent with capital M. But if you are using millimoles, then we will be getting millimoles. Okay, but always you have to keep it in liter. Now this formula is to find out unknown molarity. If you have taken let's say 0.5 moles of solute and you have taken in 500 ml of water, but ml you can't take, so you have to make it 0.5 moles of water of the solute in 0.5 liter of water. Okay, so then you can find out the unknown molarity. But if you have to say that 1 molar will always mean 1 mole of solute in 1 liter of solution. Okay, so this will be a standard 1 molarity, but for unknown molarity, if you have to find out, you need to know how much moles are given and how much concentration is given. Now question is, if you take a certain component, how will you find out how much moles is there if the concentration, the amount is given in grams? We have just found a relation between grams and moles. Let's understand how to prepare a 1 molar solution. Let's take example 1 molar NaCl, how this can be prepared. If any student wants to try this out, you can pause the video at this moment and try solving it yourself. In case you are not able to solve it, let me tell you the answer. First of all, you have to find out the molecular weight of NaCl, because depending on the molecular weight only, we can relate with the moles and the grams, right? So just now we have found out that molecular weight of NaCl is 58.5 AMU. Now if you take 58.5 grams instead of AMU, this is the mass of 1 mole. This we have already studied, isn't it? But we want to prepare 1 molar NaCl, means we have to take 1 mole of NaCl. You can't really calculate, count and add 6 into 23 molecules of NaCl. That is not possible, isn't it? Because 1 mole is also 6 into 10 to the power 23 molecules. You can't really count this many particles. Instead, if you just take 58.5 grams of NaCl in a measuring, you can measure it out on a beaker on the weighing scale, and then with this you have to add 1 liter of water. Okay, so since 1 is in solid component and 1 is liquid, so this you can add complete 1 liter. In case you are mixing liquid with liquid, then if you are taking, let's say 10 ml, then you have to fill up the volume with water. Don't add 1 liter of water here, because then the total volume will be more than 1 liter. So you have to fill up the volume to 1 liter in such cases. But here we just have to take 58.5 grams NaCl. We have to weigh it out and adding water to make 1 liter of solution. Now you have 1 molar NaCl solution. Okay. So let's practice one question, how to find out the number of moles given the grams is given. If grams is given, how to find out moles. Again, you can pause the video if you want to try it out yourself. And for the students who are not able to do it, here is the solution. What is the molarity of 0.5 liter NaOH solution if it contains 6 gram NaOH? Whenever your concentrations are given in gram, try not to use this grams directly. Try to convert it into moles. Okay. Now again, the first step will be to calculate the molecular weight. The molecular weight of NaOH is 40, that is 23 plus 16 for oxygen and 1 for water. Now 40 grams of NaOH if we take, that means we have taken 1 mole of NaOH. But however, we have not taken 40 grams, we have taken 6 grams. Here you can apply the unitary method. 1 gram will be giving you 1 by 40 moles. And 6 grams will be giving you 1 by 40 into 6. So basically you have to divide 6 by 40. Okay. So this is going to give you 0.15 moles of NaOH. Okay. Now that we have our moles of NaOH and they have also asked in how much liter. So easily we can find out the molarity. Remember the formula of molarity, which is moles per liter, isn't it? Now, how much moles we have? We have 0.15 moles. And how much liter we have? We have 0.5 liter. That is why the molarity of this unknown, you don't know. The molarity is not directly given. It is equal to 0.3 molar. This is how easy the calculations become when you know the basic concept. So, whenever in biochemistry you are solving any enzyme based questions or pH buffer related question, thermodynamics, for everything never use in grams, you're always supposed to find out in molarity. Okay. So molarity in chemical reactions, you can use the formula to find out anything, because molarity is equal to moles per liter. Okay, that means the volume, moles by the volume. If any of this component 2 are given, you can find the other one. Like for example, if you want to find out moles, okay, provided your molarity and volume is given, so you have to bring it to the other side. So by multiplying the molarity and the volume, you can calculate how much moles is there. Or the other way around, if you want to find out the volume, then you can bring the molarity here. So the volume will be moles divided by the molarity. Okay, like this you can use. The next concept is dilution. Now that you have some concentration of the solution, what if you want to dilute it? Now what happens in dilution? Water is added. Because water is added, the volume will be increasing, and if volume increases, the concentration decreases, because now you have more water, but the solid particle or the solute particle is still same in the same amount of water. Okay, so this is what is the concentrated solution, where you have taken some amount of water. Here, compared to the water components, the solutes are more concentrated. But when you add more water but not the solute, then now in a given area the solute particles are less. So this is a diluted component. Sometimes you have to calculate for that also, initial and diluted solution. What is the formula? So moles of solute are the same. The concentration and volumes are related by the following equation. For percent concentration, you can use C1 V1 is equal to C2 V2, where C1 V1 will be for the initial component and C2 V2 will be for the diluted component. You can also use same as molarity, M1 V1 is equal to M2 V2. Let's see how to do it with an example. You can pause the video now if you want to try this question by yourself. Okay, so how to do this? Right now here the concentration, it is given in percentage. That is what is mass by volume. It is given in 2%. Now mass by volume means what? Percent means what? Per 100 particles, how much it is present. That means per 100 water, how much solute is present. That is what it means. But you have to put some unit. So for water or the solvent particle, you use 100 ml. So per 100 ml, how much grams of solute is present. That is how you have to give the mass by volume. Now since you have taken 100 ml, how much grams? So 2% means you have taken 2 grams per 100 ml. Okay, here you don't need to know the molecular weight like in molarity. You can directly find out how much percentage it is there. Like this you can even have volume by volume. In case your solute component is a liquid and not a solid, because for liquid you cannot use gram, right? Now what volume of a 2% HCl solution can be prepared by diluting this much ml of 14% HCl? Whenever you have such questions, you need to know initial and final. The final will always be the higher concentration. Okay, because here you have more amount of solute, so we will have 14 grams per 100 ml. This is what means 14%. You don't need any conversion because both the units are in percentage, mass by volume percentage. Always whenever you have to do any calculations with any equation, you have to convert into the same type of units. With different units you cannot put into an equation. Okay, so 14% will be the concentration of the initial. That means this is C1, and since the volume it is with this concentration, so this will be V1. Now the lesser concentration is C2. All you have to find over here is what is the V2. That means how much volume will be prepared if you take 25 ml of this concentrated solution. So first of all, write down what is given. At this point, if any unit conversions are done required, you have to do that and put it in the equation. Easy calculation. Since you have taken the volume in ml, the answer also you will be getting in ml. But if your choices in the options are given in any other unit like liter or microliter, then accordingly don't forget to convert. Okay. Next concept is molality. This was previous one was molarity, and now this is molality. It is just an alternative concentration which are nearly as useful, but mostly in your numerical and CSIR you will be getting molarity. Molarity refers to the number of moles per kilogram of solvent. How we have used in molarity per liter, but here for molality we will be using per kg. Okay, when the solvent is also a solid component, that is where we use the term molality. These are the unit reviews. You are supposed to know this and be well versed in the interconversions. For example, if you take grams, then milligrams will be 10 to the power minus 3. Okay, so minus 3 grams. So if you take 1 milligram, it will be equal to 10 to the power minus 3 grams. If you take 1 gram, it will be equal to 10 to the power 3 grams. So minus 3 means, it means 1 by 10 to the power 3. 10 to the power 3 means 1 followed by 3 zeros. Okay, this you can also write it. So this is the one term. Since 3 zeros you are dividing, you have to shift. So here would be the invisible point that is not written over here. It's actually 1.0 also you can write. You have to shift the point 3 places. That means you have to have 0, 0 and here you can shift. So 1, 2 and 3. So this can also be written, 1 by 1000 can also be written as 0.001. These are the conversions which I am talking about. You should be very familiar with for solving numerical calculations. Okay, be familiar with the micro and nano also. Remember something, if from a bigger unit you are going to a smaller unit, then whatever is the difference, for example here from gram to milligram, milligram to microgram, microgram to nanogram. So each one of this difference is 1000. That means total difference is 10 to the power 9. If you are going from a bigger unit to a smaller unit, you can multiply with the difference, and if you are going from a smaller unit to a bigger unit, you can divide by the difference. Okay, so you can either divide by the difference or you can multiply with 10 to the power, means this difference but putting a minus. But this way it will be more easier for you. Don't confuse yourself unnecessarily. Practice more such conversions for better performance in exam. Next is percent solution, which I already mentioned to you. Solutions based on percent are the easiest to calculate because they do not depend on the knowledge of the molecular weight. If you are given a tube with an unknown white powder and you have to make a 1% weight by volume solution, you can do it without knowing anything about the white powder. Okay, so W by volume, it means percent weight to volume and has unit of grams per 100 ml. So if you take 1% mass by volume or weight by volume, it means 1 gram of the solute. Any solute you can take in a total of 100 ml of solution without having to know its molecular weight. Okay. The last topic for today is normality or normal concentration. Now here our denominator is same like molarity, volume of solution, which we'll take in later. But instead of taking just the molar mass, we have to take something called as equivalent weight, equivalent number of the solution, of the solute. Okay. So what is this equivalent number? It is equal to the weight of the solute expressed in grams divided by the equivalent mass. Okay. Now, whenever we take molarity, we take the molecular weight, the gram amount in gram divided by the molecular mass divided by liter of the volume in liter. But if we take normality, we have to take equivalent mass divided by volume. Equivalent mass is calculated by same weight, how much of solute particle is there, but divided by the equivalent mass, not the molecular mass but equivalent mass divided by volume by liter. So whenever you take equivalent weight, it will be molecular mass divided by N. Now what is N? Here I told you, you have to use the equivalent mass. So how to calculate equivalent mass? How it is related to molecular mass? Equivalent mass is molecular mass divided by N. Now what is this N? N is actually the active unit in that particular component. Okay. For example, the active unit in acid-base reaction are the number of hydrogen ions which are released by a single molecule of an acid or reacted with a single molecule of base. That is what is active unit. For example, we know acid is something which gives away H plus in water. If you use monoprotic acid like HCl, it gives away only one proton. So HCl active unit is 1. That means for HCl, molecular mass and equivalent mass is same, which means its molarity will be equal to normality. Okay. But if you use H2SO4, for this you have 2 protons to give away. So here active unit will be 2. which means the molecular mass will be the equivalent mass will be molecular mass divided by 2, or the molarity will be equal to, the normality will be equal to Nm. Okay, so this is that contains 2 protons. If you use active unit in oxidation reduction reaction, it is the number of electrons transferred from one reactant to another during the reaction. Okay, so this is how, as I said, normality will be equal to molarity into number of active unit. So equivalent mass is the substance that can be calculated for dividing its molecular mass per the number of active unit in one molecule of substance. That's all for now. Follow Biotecnika videos, and you can also enroll in the regular batch classes if you want to practice more numericals to have a strong grasp on the problems coming in the CSIR exam. Thank you everyone.